Welcome to this video on gluconeogenesis, an essential metabolic pathway. Glucose is the primary energy source for many vital organs, including red blood cells, the brain, exercising muscles, testes, and the renal medulla. When blood glucose levels drop or energy demand increases, the body maintains an adequate glucose supply through two pathways, including gluconeogenesis. In this video, I will explore the definition, location, the key precursors or substrate, the mechanism, the steps and enzymes involved, regulatory steps, and the significance of gluconeogenesis. Gluconeogenesis is defined as a metabolic pathway that synthesizes glucose from non-carbohydrate precursors or substrates during fasting, starvation, or low carbohydrate states. Regarding the site, the liver is the primary site of gluconeogenesis during early fasting when dietary intake stops. About 90 to 95% of gluconeogenesis occurs in the liver. The remaining 5 to 10% occurs in renal cortex. However, during a prolonged fasting, typically after 24 to 48 hours, the kidneys significantly contribute to glucose production. This dual contribution ensures a steady supply of glucose for vital organs such as brain, etc. So regarding non-carbohydrate precursors or substrate, the first one is alanine. This is produced in the liver as well as in the exercising muscles. It is a major contributor. Next one is glucogenic amino acids. Except lysine and leucine, the remaining all amino acids are glucogenic amino acid. They contribute to gluconeogenesis. The next one is pyruvate, is also produced in the muscle as well as in the liver. It is a substrate for gluconeogenesis. Lactate is produced in the RBC as well as in the exercising muscles and liver, and this is a major contributor to gluconeogenesis. The next one is glycerol. This is not a major contributor. This become significant in fasting state, and this is produced by lipase action on triglycerides. The last one is propionyl CoA. This is derived by the oxidation of hot chain fatty acids, and hot chain fatty acids are not present in humans. Therefore, the source of this is certain glucogenic amino acid as well as the microbial activity in the GI tract. In the next slide, we will identify the sites where they enter in the gluconeogenesis. Except for glycerol, the remaining non-carbohydrate substrates such as lactate, pyruvate, alanine, glucogenic amino acids, and propionyl CoA ultimately converge into oxaloacetate. Therefore, oxaloacetate is considered as the central hub and gateway to gluconeogenesis. Gluconeogenesis is reverse of glycolytic pathway, and therefore I would like to compare the two processes before describing the mechanism. They are related but mutually exclusive, means if one is functioning, other will be silent. Gluconeogenesis consumes ATP while glycolysis produces ATP. Next point is gluconeogenesis is reversal of glycolytic pathway, except there are three enzymes which are different. We will identify those enzymes in the next slide. Gluconeogenesis synthesizes glucose while glycolysis breaks down glucose. The next point is glucogenesis occurs in liver cell cytoplasm and mitochondria, while glycolysis occurs in the cell cytoplasm. They are reciprocally regulated by insulin and glucagon hormones. So let us identify the points of differences between glycolysis and gluconeogenesis. Glycolysis involves conversion of glucose into pyruvate, whereas gluconeogenesis convert pyruvate into glucose. So basically, gluconeogenesis is the reversal of glycolytic pathway. Glycolysis involves 10 different enzymes. However, I have shown here three enzymes only, or three steps, because these are the focus of differences between the two pathways. Except these three steps, the remaining steps are catalyzed by the same enzyme in both pathways. So the first point of difference is the first step of glycolytic pathway, which is carried out by hexokinase conversion of glucose into glucose 6-phosphate. The reversal of this is carried out by glucose 6-phosphatase enzyme. This is the last step of gluconeogenesis. This enzyme is present in kidneys and liver, and therefore these organs supply glucose to other body parts as well. While this is absent in skeletal muscle, and therefore skeletal muscle cannot supply glucose to other body part. The second enzyme which is different in both pathways is fructose 1 fructose phosphofructokinase 1, which convert fructose 6-phosphate into fructose 1,6 bisphosphate. This reversal of this is catalyzed by enzyme known as fructose 1,6 bisphosphatase. So this is the second enzyme which is different in gluconeogenesis. The third step is the last step of glycolysis, which is carried out by pyruvate kinase, involving conversion of phosphoenol pyruvate to pyruvate. The reversal of pyruvate to phosphoenol pyruvate is catalyzed by PepsiK or phosphoenol pyruvate carboxykinase enzyme. So this is the first step of gluconeogenesis. So except these three enzymes, the remaining seven enzymes are same in both pathways. So let us discuss the first step of gluconeogenesis, which is conversion of pyruvate into phosphoenol pyruvate. This occurs in the hepatocyte cytoplasm as well as in the mitochondria. So pyruvate is the major non-carbohydrate precursors, which is derived from lactate by lactate dehydrogenase. In this reaction, NADH is produced, and the another source of pyruvate is transamination of alanine. Alanine is a major non-carbohydrate precursor for the gluconeogenesis. Pyruvate enters the mitochondria and it is converted into oxaloacetate by the action of pyruvate carboxylase enzyme. This enzyme requires biotin, ATP, and carbon dioxide. Oxaloacetate basically is a central hub and gateway to gluconeogenesis. There are group of amino acid, they are called glucogenic amino acid. They finally converge into oxaloacetate and enter into gluconeogenesis. Similarly, propenyl CoA also finally converge into oxaloacetate. Oxaloacetate is reduced to malate by malate dehydrogenase. In this reaction, NADH is consumed. Malate diffuses out into cytoplasm where it is acted upon by malate dehydrogenase cytosolic isoform, which which produces NADH and also oxaloacetate. Oxaloacetate is finally converted into phosphoenol pyruvate by PepsiK or phosphoenol pyruvate carboxykinase. This enzyme utilizes GTP as a source of energy and CO2 is released. GTP is equivalent to ATP. So in this pathway, two molecules of ATP have been utilized. So this is the major pathway through malate to PepsiK. The minor pathway is through PepsiK mitochondrial isoform, which also directly produces PEP or phosphoenol pyruvate from oxaloacetate. This is the minor pathway route which joins the cytosolic pool of phosphoenol pyruvate and then participate in gluconeogenesis. So this is basically the first step of the gluconeogenesis. In the next slide, I will discuss the complete pathway. So let us discuss the mechanism of gluconeogenesis. In this cartoon, I will emphasize on 3 points. The first, the complete reaction enzymes of gluconeogenesis, and the second, I will identify the point of differences or identify the enzymes which are different in gluconeogenesis as compared to glycolytic pathway. Third point, I will identify the entry point of different non-carbohydrate precursors into this pathway. So let us start with the first step, conversion of pyruvate to phosphoenol pyruvate by PepsiK. This step I have already examined in detail in the previous slide. So phosphoenol pyruvate in the next step is converted into 2-phosphoglycerate by enolase enzyme, which is then converted into 3-phosphoglycerate by mutase. which is then converted into 1,3-bisphosphoglycerate by enzyme called phosphoglycerate kinase. Here, this step utilizes one ATP molecule. Next step is the conversion of 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate by the dehydrogenase enzyme. Here is the utilization of 1-NADH. In the next step is isomerization of glyceraldehyde 3-phosphate into DHA dihydroxyacetone phosphate. These two together forms fructose 1,6-bisphosphate by the action of enzyme aldolase. Fructose 1,6-bisphosphate is converted into fructose 6-phosphate by fructose 1,6-bisphosphatase. So this enzyme is different in gluconeogenesis. So this is the second point of difference. The next step is isomerization of fructose 6-phosphate into glucose 6-phosphate, which is finally converted into glucose free glucose by glucose 6-phosphatase. So this is the third point of difference here. So these three enzymes, PepsiK, fructose 1,6-bisphosphatase and glucose 6-phosphatase, are different in gluconeogenesis. The remaining enzymes are same in both the pathways. So glucose 6-phosphatase, as I mentioned earlier, is present only in the liver and kidneys, and therefore these organs supply glucose to other body parts. This enzyme, whereas, is absent in the skeletal muscle, and therefore this skeletal muscle cannot supply the glucose to other body part. Now coming to the the point of entry, glucogenic amino acid as well as propionyl CoA finally converge into oxaloacetate, and they enter into glycolytic pathway through PAP or phosphoenol pyruvate, whereas alanine and lactate enters into this pathway through pyruvate. The last one is glycerol, which is converted into glyceraldehyde 3-phosphate by glycerol kinase and enter into the gluconeogenesis at this point. So this is the mechanism of gluconeogenesis. Conversion of 2 pyruvate into 1 glucose molecules requires 6 ATP and 2 NADH molecules in gluconeogenesis. Therefore, the process is highly energy dependent. Gluconeogenesis is physiologically very important process because it ensures an adequate supply of glucose to the vital organs including brain, testis, renal medulla and RBCs in the fasting state. Gluconeogenesis is regulated by three mechanisms including induction and repression, covalent modification by phosphorylation, and allosteric regulations. In the coming slides, we will discuss each in detail. Gluconeogenesis is induced during fasting and inhibited during fed state by hormonal regulation of gene expression of PepsiK, fructose 1,6-bisphosphatase, and glucose 6-phosphatase, which regulate gluconeogenesis. Increase in the level of glucagon, epinephrine, or cortisol causes induction of gene expression of these enzymes, thereby inducing the activity of gluconeogenesis. The mechanism of action of glucagon and epinephrine involves cyclic AMP protein kinase A signaling mechanism, whereas cortisol acts through receptor mediated mechanism. On the other hand, when the level of insulin is increased, it causes suppression of gene expression of these enzymes, thereby inhibiting gluconeogenesis, and this act through PI3K and AKT signaling mechanism. Covalent modification of gluconeogenesis occurs by phosphorylation or dephosphorylation of key enzyme known as phosphofructokinase 2, which has two activities, kinase as well as phosphatase activity. This is under the control of hormone, glucagon and insulin. Glucagon increases the activity of adenylate cyclase, which causes increase in cyclic AMP and thereby increasing the activity of protein kinase A, which phosphorylates phosphofructokinase 2, thereby increasing its phosphatase activity that causes decrease in the level of fructose 2,6 bisphosphate. This causes or this activates fructose 1,6 bisphosphatase enzyme and therefore activates gluconeogenesis. On the other hand, insulin activates phosphatase enzymes which dephosphorylate protein phosphofructokinase 2 enzyme, thereby decreasing its phosphatase activity that leads to accumulation of fructose 2,6 bisphosphate that which decreases the activity of fructose 1,6 bisphosphatase, thereby inhibiting gluconeogenesis. So let us discuss allosteric regulation. There are two enzymes in this category, pyruvate carboxylase and fructose 1,6 bisphosphatase. Both are activated under high energy state, whereas they are inhibited under low energy state. Specifically, pyruvate carboxylase is activated by high concentration of acetyl-CoA and inhibited by high concentration of ADP, whereas fructose 6,1,6 bisphosphatase is activated by high concentration of citrate and inhibited by higher concentration of AMP and for fructose 2,6 bisphosphate. Thank you very much for watching this video. Please like it, share it and subscribe it and also write comments. Until next time, you take care, bye bye and have fun.